Good morning, everyone. We're just trying to get Pumudzo online. Pumudzo, you're muted. All right.
Can you hear me?
Yes. Welcome, everyone. I'm handing over to Pumudzo to start us off for the day.
Good day. Before we get started with the delivery of presentations, I would like to express my sincere appreciation to everyone who's attending this presentation. So I would also like to congratulate our presenters for registering their presentations on this session on impacts of biological invasions. Indeed, biological invasion may put both direct and indirect impacts which are either negative or positive to our daily lives. Certain consequences are economic, some are social, and are perceived at our environmental, biodiversity, water supply, plantations, compliance matters, and or agricultural setups. From these upcoming exhibitions, we'll hear how biological invasion affected our previous, current, and future situations. The current session consists of 9 recorded video presentations. It is important to note that while the presentation is running, questions can be addressed directly to the relevant presenter using the @ symbol together with the presenter's name on Q&A platform. The chat box platform can be used for general comments. However, I would like to encourage all the presenters and panelists to mute their video and audio controls while the presentations are underway. Let's enjoy our presentations.
Welcome, everybody. My name is Sabrina Kumschik. I am the risk analysis researcher at the Center for Invasion Biology at Stellenbosch University, funded by SANBI, and I'm also a member of the IUCN's Invasive Species Specialist Group. I'm very pleased to introduce you to the new IUCN standard for classifying alien taxa according to the magnitude of their impact. On the 15th of September in 2020, the IUCN launched a new standard to classify alien taxa according to the magnitude of their impact, the Environmental Impact Classification for Alien Taxa, short ICAT. In this presentation, I'm going to show you how this standard came about and what hoops we had to jump through to get ICAT to where it is now. The idea to get an IUCN standard for impacts of alien taxa started in 2013 at a workshop in Leipzig, Germany, with these jolly fellas here on the picture, where we discussed how alien species impacts can be quantified and predicted. One of the papers which were produced at this workshop was an impact scoring system for alien taxa. The paper was published in PLOS Biology in 2014. The classification system we proposed there is based on previous frameworks categorizing alien species impacts, and it aligns with global databases like the IUCN's Global Invasive Species Database, GISD. Many of you are probably familiar with this. Furthermore, it included a measure of confidence for the assessments. For a framework to become an IUCN standard, it needs to go through a global consultation process. Any stakeholder from the vast IUCN network and other interested parties can be involved and comment on the proposed method. In our case, 260 IUCN members and other individuals from 77 countries from all continents responded. The most responses came from India, followed by the USA, UK, and France. And the support we received for ICAT was overwhelming. Just to show you 2 examples. Green is strongly agree or agree, yellow is neutral, and orange and red are disagree or strongly disagree respectively. Only 7 respondents think they will not use results of ICAT assessments for example, to support prioritization of management actions. And also, the vast majority of respondents supported the adoption of ICAT as a standard, with only 4 of 260 disagreeing. Of course, we received a lot of comments from this consultation process, and after revising the ICAT categories and criteria based on these comments, and based on our own experience using the scheme, ICAT was adopted as an IUCN standard early in the year 2020 and launched in September 2020 at the Neobiota conference. What you see on your right-hand side are the basic categories into which an alien species can be classified. From the bottom, we have species which have not been evaluated against the ICAT criteria and species which have no alien populations, so which have never been introduced outside of their native range. Unfortunately, for many taxa that we have evaluated up to date, about a third were classified as data deficient, as no information on their impacts on native taxa was available. Then coming to the impact categories shown in the orange box. In ICAT, alien species are classified according to the impact they cause on native biota. Again, from bottom to top, minimal concern impacts are given when there is evidence that an alien species has not affected the performance of any native species. Minor means the alien species affects the performance of at least one native species, but not its population size. Moderate is an alien reducing the population size of at least one native species, and major is assigned if an alien leads to the local disappearance of at least one native taxon, and if this impact is irreversible after the alien is taken away from the system, it is assigned the massive category. Since the launch of the ICAT standard, we have developed some additional guidance documents to help people use the ICAT standard, as well as the data produced. This includes detailed guidelines, a summary of the changes made to ICAT since it was first published in 2014, guidance on how to use the data and classifications, and some clarifications regarding uncertainty assessments in ICAT. Currently, ICAT is also being integrated into the Global Invasive Species Database, and in the very near future, you will be able to download assessments for all alien amphibians from this database to begin with. And many other taxa are also currently under assessment and will be added here when they are done. But assessments do not automatically get added once they are finished. They first go through a rigorous review process to make sure that the assessments use the guidelines correctly and that they are complete. You don't need to be able to read all of this, but this is just to illustrate how the review process works. The ICAT authority, shown in blue, consists of several experts in biological invasions, which are also familiar with the ICAT guidelines. It is responsible to administer the review process of ICAT assessments. The ICAT unit in green is responsible for a final consistency check before things are published. ICAT assessments are either done within the ICAT authority or by external experts, shown in orange here. In both cases, they are submitted to the ICAT authority chair, whose function I'm currently fulfilling, and a handling authority member who was not involved in the assessment is assigned to conduct the review. This process is similar to how reviews work within scientific journals. Then the reviewer comments are sent back to the assessor, the assessment is revised, etc., until it can be accepted for publication on the Global Invasive Species Database, here shown in red. You might be thinking, now what? What is this all good for? Why all this fuss about ICAT? ICAT is way more than an academic exercise. The ICAT guidelines or protocol can be used to simply put a large diversity of impacts into a common currency, which can help us answer research questions like why certain species have higher impacts or under which circumstances they do. But of course, the data collected during such an exercise can also be of value to people making decisions on which alien species to manage or how to regulate them. Just to give you some examples from South Africa. ICAT classifications are used as one indicator in assessing the status of biological invasions in the country. They also directly feed into risk analysis conducted to decide which alien species to list under national regulations. More about the uses of ICAT can be found in this paper published in the journal Neobiology. Now, most importantly, how can you be involved? Firstly, you can be an assessor. If you use ICAT for your research, please fill it into the IUCN's template and submit it to the IUCN authority for review. This gives your research more visibility, and it helps the global conservation community by providing the evidence needed for the fight against harmful alien species. If you are not currently using ICAT in your research project, maybe consider using it. Secondly, when you do studies on impacts of alien taxa, provide the information needed by ICAT to assign an adequate category to the impact record. For example, don't just report on how alien trees affect the native soil, but also look at how native biodiversity is affected by the alien species and to which extent. Then last but not least, we are looking for funding to keep the ICAT authority and all the related processes running from next year onwards. This includes the development of a freely accessible global database on impacts of alien taxa, which can help a wide variety of stakeholders, potentially including yourself, to make better and more transparent management decisions. If you can help, please get in touch with me. Many people are and were involved in the ICAT process and in getting it to where it is now, accepted as a global ICAT standard. I would like to thank them, my funders, and you all for your attention. Thank you.
Hello, I'm John Wilson. I work for the South African National Biodiversity Institute, and I'm the science lead for their work on biological invasions. I'm also a core team member at the Centre for Invasion Biology. In this talk, I want to introduce you to the Alien Species Risk Analysis Review Panel, or ASRARP for short, and encourage you to get involved in the work it does in providing the scientific evidence to underpin the regulation of alien species in South Africa. To give you an idea of the scale of the issue, here are all the taxa that are listed under South Africa's alien and invasive species regulations, all 566 of them. I'll highlight a few so you can actually read them. So one of the first questions that comes to mind is, how did we decide which species to list? Later we might ask, can we add a species to the list, or can we take something off it? These are, of course, societal decisions made by the government on behalf of all South Africans. Specifically, it's the Department of Forestry, Fisheries and the Environment. I'm not sure of the correct pronunciation of DFFE, so I'll just refer to it as the department from here on. There have been two major updates to the lists since they were first published in 2014, but the evidence required to underpin such changes is not clearly laid out. Thinking as a scientist or manager working on biological invasions, we have a specific and important role to provide relevant evidence to support the decision makers in their job. In part to address this issue, the Alien Species Risk Analysis Review Panel was set up. ASRARP is a scientific advisory panel dealing with issues pertaining to the risk posed by alien species to South Africa. It does not have an explicit legal mandate, but there is provision in the regulations that import applications may be submitted by the department to SANBI for scientific review. This picture shows a meeting we held in 2018. And here's a more recent picture of a meeting on Zoom. To provide some more background, ASRARP is funded by the department with the Secretariat run through SANBI. However, the purpose of ASRARP is to be independent of both the department as the decision makers and SANBI as an entity of the department. It is this independence that provides ASRARP its legitimacy of being able to provide scientific advice and oversight of processes. ASRARP's inaugural meeting was during November 2016, though initially government officials were part of the meetings. It has been in its current independent format since August 2018 and has met quarterly since. Members have a range of expertise and come from universities, research institutions, and private consultancies, but they all act in their personal capacities. There are 4 ex officio SANBI members, 2 of whom are observers. But speaking from personal experience as one of the ex officio members, while we can provide the terms under which ASRARP operates, we can't dictate to them what they think. We're really pleased to have a strong current chair in Lynn Jackson, and she, together with the other 19 independent panel members, have done much to build ASRARP's credibility. ASRARP has 3 main functions. First, to review applications to import alien species. Second, to review risk analyses on alien species. And third, to provide independent scientific advice on other issues. For the rest of this talk, I'll concentrate on the second one of these functions. So in terms of the process to review risk analysis on alien species, the stakeholder or the assessor will produce a risk analysis report in a set format and either submit directly to SANBI or to the department, who may then ask SANBI to conduct a scientific review of it. SANBI will conduct some internal quality control and may ask for revisions at this stage. Then the risk analysis report will be submitted to ASRARP. Such risk analysis reports may recommend changes, for example, listing new alien taxa, delisting taxa, or changing listing categories. But many, over half to date, have recommended that the listing does not change. ASRARP solicits at least 2 external reviewers for each risk analysis it considers, with working groups like the Cactus Working Group potentially acting as one of those reviewers. The review comments are then discussed at a meeting of ASRARP, with feedback consolidated and sent on to SANBI, who communicate that with the assessor, and the process may continue like this. Once ASRARP has approved a risk analysis report, they send it to SANBI for submission to the department. The department is in the process of setting up a decision-making panel which will consider the recommendations along with other stakeholders involved. As needed, the department will publish a proposal to revise the list for public comment, and following the process, potentially change the lists. This process is still bedding down, and so the regulatory lists to date have not as yet gone through the process. However, the goal is for all listed taxa to have risk analyses conducted on them, and for all changes to the regulations to be supported by risk analyses in future. The framework used for risk analysis reports was developed based on adapting international best practices and fundamental principles in risk analysis to the South African context. The framework was published in 2020 in the journal Neobiota, and the latest version and templates will always be available at the link at the bottom of your screen. I'll show it again at the end of the talk. So here's an example. Capybara is the world's largest rodent and arguably a keystone species in parts of South America. The risks are not well known, but there was sufficient information to score them as high. It is currently listed as Category 2, meaning that permits can be issued to keep the species, but few permits have been issued and the benefits are unclear. It's possibly used for hunting. As a result, the risk analysis recommends to list the taxon as Category 1B, noting that appropriate institutions like zoos can still apply for permits for display purposes, given the value of capybara for educational purposes. But the evidence that it is still in the country was very weak, and so the recommendation was to reassess the species in the next 5 years, pending confirmation of whether it is actually present in South Africa or not. If you know that capybara are around, please let us know. To date, risk analyses have been completed on around 10% of all listed taxa. However, the process is up and running and more risk analyses are coming through the system. The process is similar to an academic journal and takes between 3 and 7 months from first submission to approval. However, several have taken much longer. This is partly as the processes and protocols were not in place, but also due to delays from assessors in revising their risk analyses and reviewers in responding to review requests. The risk analyses submitted by SANBI to the department are not currently open access. This may change once the governmental decision-making body has had a chance to discuss the issue. What we have done, however, is set up a system whereby reports are submitted to Zenodo so that they have a permanent digital object identifier. This means any changes made will be traceable. To facilitate the production of risk analyses and to teach assessors about risk analysis concepts more generally, 8 risk analysis training courses have been held. If you haven't attended a course already, we would love to have you on one in future. We will plan to hold around about 3 to 4 per year from here. The theoretical part takes roughly a week, but additional time is required to do and revise a risk analysis on a chosen taxon. The focus is on scientists and senior postgraduate students, but we would strongly encourage policymakers to also attend to get an insight into the process. So how can you help? First, please get involved in developing risk analyses. SANBI will be advertising several contracts to conduct risk analyses. These contracts will be to work on species prioritized by the department. But we also want to provide a route for people to submit risk analysis on taxa they are working on. So please consider going on the training course. Second, when asked to review a risk analysis, please do so promptly and engage if a risk analysis is tabled at a working group. I know how many requests people get to read this or comment on that, but please do try to engage. These need to be our regulations. Finally, we'll be advertising for new panel members towards the end of 2021. Please consider applying. So thanks to all the people listed here who have been panel members, external reviewers and assessors. The full list is available on SANBI's website. Finally, thanks also to the Department of Forestry, Fisheries and the Environment for the funding and their continuing strong support for ASRAP as an independent panel, even when ASRAP has created work for them. Please feel free to contact me or any of the other people listed here if you have any questions. All the contact details and links are available on SANBI's website. Thank you.
Good day, everyone. Today I'm speaking under the title, The Influence of Invasive Opuntia ficus indica on the Livelihoods of the People in Limpopo Province. Opuntia ficus indica is one of the globally distributed species that was introduced in South Africa in the late 1800s, and to date it has colonized all provinces, precisely because of the benefits that are associated with these species. In this study, we asked the following questions. What are the factors predicting the trade with Opuntia ficus indica? Does experience and the distance to collect explain the income generated? And are there any gender differences regarding the people involved in the trading? The study was conducted in the 3 municipalities, and the study protocols was approved by SMU Ethics Research Committee. And out of the study, we found 72 traders involved in this trading. Majority of those were males. And as expressed by the traders, it is not a safe kind of business for females to be in the field alone. And most of these were participating in this trading because they were unemployed. And here we looked at the distances that they traveled to collect and also to the trading posts. We found that the traders along Solomon Dale walked the furthest to the selling point, and overall all selling points traveled the same distances to collect, and also similar distances to the selling post. And here we're looking at the transportation mechanisms that they use to transport the fruits. Out of the 72 traders, only one trader was found to possess a car that they use in this trading business. In this slide, we're looking at the gross seasonal income. The trading season starts in December, January, and ends in February. So we see the traders along R71 in Nobody generated the most income, up to 15,000 rands. And for this group of people who live below the poverty line, such an income is a huge income for them because that's the only income they have. And here we looked at the factors that could predict the income, and we found that age, as well as the distance to collect, does explain the levels of incomes that are generated per season. This means that the traders who are willing to walk the furthest are likely to generate more income as opposed to those who can only walk to a certain point. And we also found a positive relationship between the time the trader has been involved in this business, as well as the gross income that they are likely to generate per trading season. And here are the voices of some of the traders. One trader had this to say: I have been selling the prickly pears for more than 10 years. I have built a house. I'm taking care of my children. And I bought a bucket too from this business that I use back in this business for trading and collecting the prickly pears. With that, we can conclude that Opuntia ficus indica contributes to the livelihoods of the people in Limpopo positively. And there is a saying that if you can't beat them, you may as well eat them. Thank you.
Good morning, everyone. Thank you for the opportunity. As introduced, I'm Panelist Bomislavisa, and my project was about ant diversity and assemblage composition in relation to the invader Parthenium hysterophorus and its biological control in South Africa. Parthenium hysterophorus is an invasive weed which has spread through countries around the world, South Africa being one of those countries. In South Africa, it has invaded and spread in provinces such as KwaZulu-Natal, Mpumalanga, the Northwest, and Limpopo. It is known for its multiple impacts on ecosystems, this including its impacts on the biodiversity of flora and fauna. Several studies have reported on its impacts on fauna. However, very few studies have looked at its impacts on invertebrates. Five agents have been released for the biocontrol of this invasive weed. However, our study focused on one agent, the Listeriosus cytosipenis, the stem-boring weevil. The study aimed to investigate the effects of Parthenium hysterophorus and biological control agent Listeriosus cytosipenis on ant diversity and assemblage composition. One might ask why we focused on ants and not other invertebrates. And this is mainly because ants are known as biological indicators of environmental change, and they were observed in Parthenium plants. The study was conducted in several sites in KwaZulu-Natal and Mpumalanga provinces. In each site, a 15-meter transit was set up in 3 treatments: no Parthenium, one with Parthenium only, and one with Parthenium and the biological control agent Listeriosus cytosipenis. 5 quadrants were placed 5 meters within the transects to avoid edge effects, and at the center of each quadrant, a pitfall trap was placed. Vegetation variables such as vegetative cover, plant height, flower score were measured. We also counted Listeriosis oviposition, which is the number of Listeriosis eggs that are deposited on the flowers and covered with the frizz and are essentially noticeable. The pitfall traps were half filled with propylene glycol and left out for 48 hours. Ants collected from the pitfall trap were sorted in the lab and stored in 70% ethanol. These were further identified to species level and counted. The results showed that in KwaZulu-Natal, species richness was higher in the no Parthenium treatment, shown here in blue. But in Mpumalanga, species richness was marginal between the three treatments. The results also showed that in KwaZulu-Natal, species abundance, which is the number of individuals, was higher in the treatment with Parthenium and the biocontrol agent, shown here in gray. However, this was different for the Mpumalanga results, which showed that species abundance was higher in the treatment with no Parthenium, shown here in blue. The NMDX plot showed that most similarities in species composition between sites were observed in the treatment with Parthenium only and the treatment with no Parthenium. The similarities were also observed in treatment with no Parthenium and treatment with the Parthenium and biological control agent. It also showed that most similarities in species composition of ants in Mpumalanga were observed in treatments with Parthenium only and with no Parthenium, and also treatment with Parthenium only and the Parthenium and the biological control agent. To summarize, higher diversity was observed in areas with no Parthenium infestations and no agent in both regions, KZN and in Mpumalanga. But for abundance, higher abundance was observed in areas with Parthenium and the agent for KZN. However, in Mpumalanga, a higher abundance was observed in areas with no Parthenium. Similarities in species composition in areas with no Parthenium and Parthenium but no agent were observed in both regions, which is to say that the presence of Parthenium infestations and the presence of a biological control agent influences ant diversity and assemblage composition, with some effects differing between the regions. In future, we would like to investigate vegetative variables that may influence the ant diversity and assemblage compositions. Those may be the vegetation cover, plant height, and the flower score. We'd also like to assess adaptive behavior on Parthenium meat and also assess the potential biological control agent predation by ants. Thank you.
Good morning, everyone. My name is Lungile Kuzwayo. I am an MSc candidate at the Center for Statistics in Ecology, Environment and Conservation at the University of Cape Town. The title of my study is Detection and Mapping of Invasive Alien Plants in the Western Cape Mountain Catchments. Globally, natural habitats and human communities are under threat from rising numbers of destructive invasive alien plant species. The Cape Floristic region is South Africa's most invaded land region in terms of woody invasive plants. Invasive alien plant species are species whose introduction or spread outside their natural distribution threaten biological diversity and may cause economic or environmental harm. In order to handle ongoing invasions effectively, it is important to track the spread of species on a regular basis, and there is an urgent need for new tools and techniques to enable prompt, rapid, and accurate monitoring. Early and fast detection is needed to make the management cost-effective. Satellite-based remote sensing has emerged as an important technology for spatially mapping the occurrence of invasive species in space and time. Satellites such as WorldView 2 have been used successfully in the past to map invasive alien plant species. However, commercial satellites do come at a high cost. With the new era of Sentinel missions and multispectral data, high spatial and temporal resolution for the entire world is now freely available. Sentinel-2 has brought up the potential for land cover mapping at a fine resolution. The aim of the study is to detect and map the invasive elephants in the Western Cape mountain catchments through developing methods to discern invasive trees from indigenous vegetation, as well as building a repeatable workflow for classifying alien vegetation and running a time series to look at spread rates over time. And then finally, estimating the water flow reduction in the riparian ecosystem of the Western Cape mountain catchments through hydrological modeling. This study makes use of Sentinel-2A images. I've created 14 land cover classes, which include alien tree infestations. I'm using the random forest classifier. I have more than 4,600 points, which vary per land cover class. And then of the total training data, 80% is reserved for the training set, and then the remaining 20% is used for the validation set. This slide shows all the land cover classes from indigenous vegetation to all the alien tree infestation classes. The study area includes the mountain catchments that are found within the Western Cape water supply system, including all of the major dams. Displayed on the screen is the Google Earth Engine script for my classification model. As I've mentioned before, I'm using Sentinel-2A images from the Copernicus program, and I've filtered the dates for my study into a one-year period. Displayed on the screen are the results for my Google Earth Engine classification model. On the left of the screen is a raw unfiltered Sentinel image, and then on the right of the screen is a classified image. Please refer to the top left-hand corner of the screen for the legend, which will guide you about the different land cover classes. Currently, the overall accuracy is at 85%, which means that the classification has been quite successful in picking up the alien tree infestations against the indigenous vegetation. Again, if you refer to the legend, you'll notice that the indigenous vegetation is indicated by land cover classes with different shades of green. And then the alien tree infestations are indicated by brighter colors such as red and maroon and the purples. So you can clearly see that the model is able to pick up the alien tree infestations. On the way forward, I've completed the majority of the time series analysis. I'm currently busy with the hydrological data modeling and classification section of my study, as well as data analysis and report writing. Thank you.
Good afternoon, ladies and gentlemen. I am Willem de Beer. Rudy Paap, Mike Wingfield, and Francois Roots are the co-authors on this presentation. The polyphagous shuttle borer, or Uvulaceae formicatus, has been introduced into the country from Southeast Asia. The beetle is about 2 millimeters long, and together with its fungal associate, Fusarium uvulaceae, which it cultivates as a food source, it has the ability to kill several tree species. It was first found in South Africa, infesting trees in Pietermaritzburg in 2017. But subsequently, an unpublished Bold report was found online that confirmed that the beetle was already present in the country as early as 2012. PSHB establishes galleries in reproductive host trees, where the fungus then colonizes the wood and serve as food source for the beetle and its larvae. The fungus then kills the sapwood and susceptible host trees, and this leads to branch dieback and tree death. Galleries can also weaken the branches. PSHB affects a very wide range of trees across various landscapes. In the economic sector, it poses a risk to certain crop trees like avocados and also to wattles in plantation forestry. However, to date, it has not been detected in any commercial settings in South Africa. Natural ecosystems are also threatened, and so far 17 indigenous species has been recorded as reproductive hosts, including species of Virgilia and Erythrina. Urban forests are currently the most severely impacted environment, with 20 alien tree species reported to date as reproductive hosts. These include widely planted amenity tree species that are rapidly killed, like oak trees and box elder. Beetle fungus complex has already caused the death of thousands of trees in urban environments in South Africa, particularly in Sandton, George, and Knysna. PSHB is now well established across the country, and its presence has been confirmed in all the provinces except for Limpopo. Without assistance and in the absence of strong prevailing winds, female beetles have been reported to fly up to about 2 kilometers. Its spread is further facilitated by the presence of reproductive host trees in the landscape, for example, cluster beans or oak trees. But especially concerning is the ease of an anthropogenically mediated long-term dispersal, particularly by the movement of infested wood. With a substantial portion of the population reliant on wood for cooking and heating, firewood is a very challenging pathway to manage. Infested wood that is brought into campsites enhance the risk of spreading the beetle to natural environments. The ease of spread and wide range of host trees across sector presents many challenges with regards to its management. Various life history traits like galleries deep in the trees make the beetle difficult to control. With its most severe impact in urban environments, it became clear that many municipalities were not adequately equipped to respond effectively to this pest. For any chance to reduce the impact of a pest of this magnitude, a coordinated national response is required. The first step was to declare it as a quarantine pest on agricultural hosts. The recommendation for listing it under Category 1B in the NEMA legislation is still under review. The National Invasive Shuttle Borer Strategic Framework was developed for and recently endorsed by the FFE and DALREG. The two major components of the strategy are to support municipalities in the removal and safe disposal of heavily infested reproductive hosts, and to maintain and increase the awareness campaign among stakeholders and the public. To underpin management strategies, extensive monitoring and scientific research are ongoing at various universities under the banner of the PSHB Research Network. Thank you.
Good morning, everyone. My name is Sean Fell. I'm an MSc student at the University of Pretoria, and today I'll be discussing the impact of the polyphagous shuttle borer on pecan trees in South Africa. The first report of PSHB on pecan trees occurred in 2017 in the Jan Kempdorp area in the Northern Cape, where both the beetle and the fungus were identified as Eulaceae fornicatus and Fusarium eulaceae based on DNA sequencing. Since the first report of PSHB on pecans, we've also identified a second location in South Africa, Nelspruit and Mpumalanga, where signs of PSHB attacks have occurred. However, the severity of attacks in the Northern Cape is far greater than that at Mpumalanga. The introduction of the fungal symbiont Fusarium eulaceae is of great concern, especially to the pecan nut industry, as it is a known fungal pathogen that causes Fusarium dieback on hosts and in severe cases actually lead to tree mortality. Since the first report of PSHB in South Africa, FABI has identified 114 host trees, of which 37 are reproductive hosts. This led us to our aim to assess what the risk would be of PSHB on the pecan nuts in South Africa, and to do so, We need to determine the extent and the seasonality of PSHB in this area, confirm the identity of PSHB and its fungal symbionts on the pecan trees, as well as monitor the symptoms, progression, and impact the fungi have on the pecan trees. So what has worked for us so far? We completed a 1-year pathogenicity trial testing both Fusarium ulacei and Graphium ulacei on several pecan cultivars, and our findings from this suggest that pecans aren't at risk for Fusarium dieback, and most likely have a defense mechanism against the spread of Fusarium ulacei. We also needed to determine this, the extent and seasonality of the beetles. In order for us to do so, a geographical survey was conducted of the area. And during this survey, we came across the agricultural high school, which had several London English oak trees completely infested by PSHB. This led to our hypothesis that this agricultural high school actually acts as the source of infestation throughout the area. We also had to look at what trap design would be most efficient within the Jan Kempthorpe climate. And for this, we looked at 4 different traps: bottle trap, lingering trap, yellow bucket trap, as well as black panel trap. All 4 traps were baited with several lures to attract as many PSHB as possible. Our findings suggest that the black panel trap is the most efficient trap in the Jan Kempthorpe area climate, and that the cruciferal lures actually only remained active and effective for a maximum of 8 weeks. So once we got all this information, we could set out for trapping to determine the spread. Based on this, we used the agricultural high school as the epicenter, creating 5 transects from that, with 10 traps in each transect, just to determine how the beetles actually spread from this agricultural high school. After 8 months trapping period, our findings suggest that the activity of the beetles increased as the temperatures are higher. And as we entered the winter periods, the activity of the beetle actually decreased with the decrease in temperature. It is also important to note that the majority of activity occurred around the epicenter and not around the outskirts of the area. There were several traps on the outskirts that produced PSHB, but this can most likely be explained by 2 London plane trees infested with PSHB acting as a reservoir for attacks in that area of the area of Jan Kempthorpe. The current management strategy we have thus far is to report and remove infested trees and debris from the areas, especially that of non-crop reproductive hosts such as your castor bean, English oak, and London plane trees. The majority of the cases, the infested material is either burnt or solarized. The knowledge gaps we have is we still do not know how many generations the beetle has per year. We also don't know what the long-term effect would be on the pecan trees. And one of the most important questions that we have to ask is, will pecan trees actually become reproductive hosts over a long period of time? So with this, I'd like to acknowledge Harbus Twe and Ivan Schubach from SAPA, who helped us in field, as well as Annette Swanepoel from the Northern Cape Department of Agriculture, Land Reform and Rural Development, for helping us with our GIS mapping of the area, and Prof. John Allendale and Dr. Nicolette Taylor from the University of Victoria for giving us access to the pecan orchards on the experimental farm. With this, I say thank you.
Good afternoon, everyone. My name is Quadri Abelade Anibaba. I'm a PhD student at the Institute of Dendrology, Polish Academy of Science. I'm here to give a talk on land use is more important predictor of native abundance and diversity than invasive Titania diversifolia, and evidence from a typical tropical savanna landscape in Nigeria. Titania diversifolia is a known invasive plant across the tropical and subtropical region in the globe. It is native to Mexico and Central America, and it has been found to be cultivated or naturalized in Africa, North America, and Asia. In Nigeria, it has shown presence in farmland, roadside, river courses, and open spaces. And it has been found to form a dense monospecific growth through its aggressive colonization. Studies have looked at its distribution in Nigeria. However, none of these studies have looked specifically on its impact on native species in the context of land use types. Here we aim to assess the abundance of Titania diversifolia and its relationship with native species across 3 study land use types, that is roadside, riparian, and farmland. We hypothesize that land use will support a comparable abundance of Titania diversifolia and also we also hypothesize that abundance of Titania diversifolia will be negatively correlated with native abundance and diversity across our study land use types. Here is the distribution of our study plots in the study area, just metropolis, not central Nigeria. For data collection, we distributed a 15 10 by 10 plot along 5 transect, which shows considerable establishment of Titania diversifolia. In our study plot, we identified and counted individuals of vascular plants. Authentication of native plants were done using these 2 databases, that is the Plant of the World Online and Kabi Invasive Species Database. Analysis of our data was done using R, and also for our first hypothesis, we fitted a linear mixed effect model in order to investigate if there is a difference in abundance of Titania diversifolia across our study land use types. For second hypothesis, we fitted a linear mixed effect model to determine if the presence of Titania diversifolia explains native diversity. And lastly, we fitted a generalized linear mixed model to evaluate the effect of Titania diversifolia on native abundance across our study land use types. For our first result, we found out that there is no significant difference in abundance of Titania diversifolia across our study land use types. However, farmland shows more presence of Titania diversifolia, and this could be attributed to various anthropogenic factors such as land clearing and intense fertilizer application that is being done on farmland. Also, cultural practices such as hand weeding would also promote the continuous establishment of Titania diversifolia on farmland. The effect of Titania diversifolia on native diversity we don't find a significant difference across our study land use types. However, the land use characteristics could be a significant predictor of native diversity. Here, we found out that riparians still support high diversity of native plants when compared to farmland and roadside. For abundance of native species with respect to abundance of Titania diversifolia, we found out a decline in native species with increase in Titania diversifolia on farmland, which however, for other land use, the riparian and roadside, we found a different behavior of native abundance in the presence of Titania diversifolia. And this could show that farmland is highly susceptible to plant evasion. And also the high presence of nutrients and disturbance in farmland could be as a result of, could be the reason for the decline in native abundance. Our study have showed that disturbance in these land use types has resulted to abundance of Titania diversifolia and in turn reduce native species. And our findings also give a considerable evidence that the anthropogenic disturbance in sub-Saharan Africa is promoting alien plant species evasion and native species decline. And as such, we propose that for us to have a considerable management of Titania diversifolia, there is need for conservation, synergy of conservation efforts, such as preservation of our landscape and reducing disturbance on our landscape. This will help to curtail the spread of Titania diversifolia. I would like to appreciate these following individuals. Thank you for listening.
Hello, good morning, everyone. My name is Antonella Petruzzella. I'm a postdoc at the Center for Biological Control at Rhodes University. And today I'm going to present some results of a project called MADMAX, Mass Development of Aquatic Macrophytes, Cause and Consequence of Macrophyte Removal for Ecosystem Structure, Function, and Service. MADMAX is an international collaborative project funded through the 2017 call of the Water Challenges for a Changing World, known as Water JPI. It includes 5 partners, us here from South Africa, Norway, Germany, France, and Brazil. The mass development of aquatic macrophytes in rivers, dams, and lakes is a worldwide problem, and considerable resources are spent every year on macrophyte removal. What about the benefits? Macrophytes, including very dense stands, also have positive effects on aquatic ecosystems. Unfortunately, most ecosystem services provided by these plants are largely unknown to the public and water managers. Consequently, management decisions are based on prevailing negative perception of macrophytes rather than a rational knowledge-based decision. Therefore, we ask what the actual effects of macrophyte presence or their removal on structure and functional diversity of aquatic biota and water quality. These are 2 of many questions that we want to answer with MADMAX project. The MADMAX project sampling in South Africa was conducted at Copperholt at the Hartbeespoort Dam. This site belongs to the Department of Water and Sanitation and is heavily invaded by Potendera craspis, or most commonly known as water hyacinth. Our sampling was performed during early January 2020, before the pandemic hit South Africa. Here, our sampling approach followed a back design before-after control impact, whereby 2 sites were selected and allocated either control or impact. The core of sampling in back design is 1 week before and 1 week after water hyacinth removal, in order to make sure that the case studies are comparable. A variety of measurements were taken, but I'm going to focus here on a few biodiversity and water quality measurements. Here we can see the amount of biomass, grams dry weight per square meter, present in each sampling site before and after water hyacinth removal. At the control site, biomass remained constant during the experimental period, ranging around 1,000 grams per square meter. However, in the impact sites, the removal removed 1,289 of water hyacinth dry weight per square meter, which is impressive. Regarding the macroinvertebrates diversity, in total, we identified 25 macroinvertebrate families. Oligochaete was the dominant family, which is typical of organically polluted sites like ours. We calculated Shannon's diversity index values for both sampling sites before and after water hyacinth removal. Error bars in the graph represent standard errors. We found that diversity of macroinvertebrates decreased after the removal of water hyacinths. Using electrofishing, we found a total of 8 different fish species in the open water of the dam. They were caught outside of the control and impact sites. The sites covered with dense water hyacinth appear to have less diverse and abundant littoral zone dependent fish species. We only found 3 species. It's also likely that the dense coverage of water hyacinth provides a homogeneous habitat with little open water and light for many species. The exotic Gambusia finis dominated the catch. After we removed the water hyacinth, we observed a higher number of fish species. Water quality in terms of phosphates is poor at the Harveysport Dam. It ranged between 1.5 and 3.3 milligrams phosphorus per liter for both sites. The international standard is 0.1 and the South African standard is 0.2 milligrams phosphorus per liter. The water hyacinth removal seemed not to have affected the nutrient concentrations in the water. Therefore, the removal of water hyacinth had a positive effect on the biodiversity of fish, but not for the biodiversity of macroinvertebrates. Also, it did not affect phosphorus concentrations in the water. If anyone wants to know more about the project, visit our website. It's here in the slide. I'll be happy to answer some questions. Thank you very much.
Thank you very much to all our speakers for the very informative presentations. We have quite a lot of questions for discussion time here in the question and answer box. So let me get that going for us. Just checking that all of you can hear me. Looks like it, no complaints. All right, let's get to it. I'm going to start with, let's see, first question for Claude Moshabani. Are the traders aware that the prickly pear is invasive and should be controlled? And do the traders feel that their livelihood might be affected by these management options?
Okay, thank you, Yolanda. We found that the traders were not aware that these are invasive. They were not even aware that these are alien plants. Of the 76, of the 72, only 3 were aware, knew that there's the word alien, and 2 of them only knew that from their textbooks. It's only those who were school going, and the other trader only knew the word alien from the movie that is called The Aliens. All of them actually didn't know about alien and invasive species. Therefore, they wouldn't even know that they require management. They wouldn't even know whether this management of this Opuntia ficus would affect their livelihoods.
Thank you, Claude. Let's move on to a next question, which is posed to Willem de Beer with regards to the polyphagous shuttle borer. It's a question from Guy Preston with regards to what the progress is in researching any biological control agents for the management of the shuttle borer and its fungal pathogen.
Thank you, Guy, for the question, a very pertinent question. As we've got substantial funding for this biocontrol work, we have actually appointed a technician who started in the beginning of this year full-time on this work. And we just recently found, together with Francois Roots, some parasitoids on shuttle borer infested trees, which has really looked promising. So we've got some of those going in the lab. Unfortunately, in terms of the postdoc that we tried to appoint for this is a very, very competent lady from Cameroon. We've battled for 6 months now to get her into the country, and the visa application was just rejected yesterday. It just seems as if they're moving the goalposts all the time. So we, and I can announce it here, we're really looking for a competent postdoc to go full-time onto the biocontrol work. But at least the work has started. We've got serious delays with the COVID stuff, but we're happy to say that we're making progress there. And we also go together with that, the surveys in the natural forests in Tsitsikamma and Durban area is continuing. And at the same time, we are also looking for biocontrol agents there while we're looking at the impact in the natural forests.
Thank you, Willem, and that's great news and a relief to all of us, I think. Next question is to Fanelisiwe. How did you account for the differences in environmental conditions across your treatments? Fanelisiwe, you're still muted. There we go.
Hi, thank you. Okay, so far we did not account for the different environmental conditions, but with the data collected in different regions, which receive different kinds of rainfall and different temperatures, we could also use the rainfall and temperature data to account for whether the treatments had pathinium or no pathinium at all, and the agent.
I trust that answered the question. Next one is to Quadri. Quadri, this is a question from Martin Hill with regards to the impact of Tithonia in Nigeria. He's asking or wondering, would the country consider biological control against this invasive?
Yes, thank you very much for the question. Looking at the impact of Tithonia diversifolia in Nigeria, I cannot specifically say if the country will consider that, but however, the locals have utilized the plant for a lot of uses, like for firewood and also for medicinal purpose. So in terms of biological control, it could be a possible option, but it's left for the country to accept that. And also knowledge about biological invasion is pretty new in Nigeria. So it's a tricky thing, yes.
Thank you, Quadri. Next question then that I'll move on to, which is for Antonella. From Andrew Turner, do you think there is scope for reintroduction or boosting of the indigenous aquatic macrophytes after water hyacinth removal?
Yeah, for sure. And now it's exactly what I've been working on during my postdoc here in South Africa. We want to, let me start from the beginning. So what we've been experiencing here in South Africa is like we remove these floating plants and the systems have been reinvaded by submerged species. So we are not solving the problem. So what my postdoc is about is to try to bring back the native macrophytes and the native community to start to increase the resistance and the resilience of these systems to potential invaders. That's answered.
Thank you very much. Let's see.
Thank you.
Question here for Lungile. Lungile, if you're there. Question from Geeten Singh is, you showed your overall accuracy. What was the range of producer-user accuracy for your tree species?
I'm sorry. Good morning, everyone. Thank you, Johanna, for your question. Please, can you repeat the question?
The question is, states that you showed your overall accuracy. What was the range of producer or user accuracy for your tree species?
Oh, yes, I wanted to add that slide, but we ran out of time. So the range for the different classes range from 68 to about the max, which was 85. So most of the classes did behave good though, especially the invasive classes.
Right. Thank you very much.
Thank you.
We're running out of time for questions, so I'm moving, and there's quite a few more, so let's move on and we'll get back to some of those if there's follow-up questions potentially. A question for Sean Fell with regards to the shot-hole borer on pecans in South Africa. You mentioned that it was picked up in 2 areas, that in the one area the severity was higher in the other. And then you presented some results with regards to inoculation studies which showed that potentially the threat is not so high to pecans. But can you elaborate a little bit on the severity on pecans that you've seen so far in those 2 sites?
Good morning. Yeah, so what I meant by the severity in the Northern Cape actually higher than that of Mpumalanga is that when we did the survey in Mpumalanga, we only came across a handful of trees that actually showed signs of 1 or 2 beetle holes, whereas compared to that of the Jan Kempdorp area, where majority of the trees within the orchard would have signs of entry holes. But it's also important to note that the entry holes would be limited to only 1 or 2 entry holes per tree. And it looks like the beetles would then leave out that tree and skip to the next tree. So the severity refers to the amount of trees that actually have been attacked, but not showing real disease symptoms on those trees. So I don't know.
Yeah, that's good. And I think that's a big relief to the pecan growers.
Yes.
Thank you very much. You'll see, many of you will be aware in the question and answer box, there's a number of questions posed, some of which have been answered, but I'm just going to highlight 1 or 2 or give the presenters an opportunity to answer them live also, to save some time for people who don't want to read them. So the first one that I'm going to repeat from the question and answer box is to Sabrina, and it's from Diana with regards to her experience with national specialist groups attached to the Survival Species Commission of the IUCN and the Caribbean, and the procedures involving the review of taxa sheets, which he states are often complex and long, especially for the global South countries with lots of limitations and resources, capacities and recognition. And she's asking, how are you planning or imagining to make this more effective and extend the capacities to do ICAT analysis?
Thank you very much for that question. Yeah, so we have found problems, or we have encountered problems finding people to review assessments and do assessments. But first of all, as opposed to the red list, these species are not, these assessments are not local, they are global assessments. So if you have an assessment for any alien species, you should have all the information from all the alien ranges in the same assessment. So you won't have to do an assessment for each country. So that reduces the amount of assessments that will be done. But also they are different to the red list in as they only use evidence that's available and published, or that can be cited. And all this evidence needs to be provided. So what a reviewer needs to do is they need to look at what is there, is that to their knowledge complete, or is anything missing, and is what is there assigned correctly to the ICAT categories and criteria. So it is less work than looking through a red list assessment, which includes a whole bunch of different kinds of information and expert opinion and formulas and analyses and things like that, which ICAT does not include. So it should be easier to review those.
Thank you.
Yeah.
Maybe just following on that, that's part 2 of the question there is with regards to, from your presentation, it seems that you didn't have much response from African countries, for example, and also maybe South American countries. And have you had any follow-up with them to try and involve them more in the process?
We did not have follow-ups with specific countries or agencies on the process. But we will definitely try and involve them more in future. For example, doing training courses for specific regions where we know the system is not used as much or isn't known that well. So for example, we had a training course in Brazil. Of course, Brazil was one of the countries that responded, but yeah, generally South American countries are sometimes less involved in these things. And so the Brazilian government actually asked for a course. And we are hoping to do that in other regions in future as well.
Thank you very much, Sabrina. I'm then going to also repeat one of the questions from the Q&A box for John Wilson, that he can explain it out loud. Also, there is this is a question with regards to assessments conducted by the provinces versus the national assessments for these alien species. So the question relates to, do you see any role of ASROB in reviewing risk assessments conducted by provinces for alien species? Because the Constitution allows provinces to implement their own risk assessments. John, if you could elaborate on that, please.
Sure. Hi, Yolanda, and thanks, Ian, for the question. So ASROB hasn't to date reviewed documents on behalf of provinces, and it also doesn't review scientific permits, specific permit applications other than import ones. Over time, ASROB might be able to assist in these sorts of roles, but there's about 500 regulated species that need a risk analysis, and there's quite a number of broad issues that need to be resolved at a national level. So I suspect we don't have capacity in the immediate term. What I would suggest is it's something that could be raised with the department and the decision-making body once that's set up. And maybe they could discuss that and see whether that's a role that ASROB could play in future. Though I would also note there's currently a bit of a gap between the costs of scientific review and how much permit applicants are charged. So there's a few issues there to resolve, but in principle, obviously, it'd be very good to be able to assist wherever we can.
Thank you, John. I'm going to follow that up with one more question, which you've answered, but just to get it out live, also because there's a lot of interest in these risk assessments from various people. So the one question here from Lulama Madire was, what is the cost of a risk analysis course per individual or group?
Yeah, so to date, we've only really had SANBI affiliates. We've been testing the course and getting it to work, and then we're beginning to open it up to all of you, the invasion science community, invasion management community. So there isn't a cost at the moment, and there probably wouldn't be a cost in terms of departmental funded work that going in. Down the line, if we're starting to getting private individuals involved, we probably will need to look at costs of attending things. But yeah, if you put in I put the email into the chat. So if you specify who you are, you're working on a departmental funded project or a government funded project, then yeah, at present we haven't got charging, but that might be something that we need to think about going forward, and making sure that the course is accredited and all those sorts of bureaucratic steps that as an academic I've been avoiding, or all of us have been avoiding. Sorry, I'm not the main person involved in the course.
Thank you so much, John. Then I see Claude is typing furiously here. I was going to address this next question and give him the opportunity to answer live also. So Claude, if you stop typing, then you can answer live. It's a question with regards to rural women. The question is from Naweji Patembu, who states that rural women are always out in the field collecting firewood and fetching water. So why is security being raised in your questionnaire with regards to Pantia collection in Limpopo?
Thank you for that question. In this case, the prickly pear trading is currently not regulated. Therefore, anyone can start trading, just go to the field, collect and start trading. Therefore, there are no impediments or exclusions that would come to females as a result of males because there is money involved. In fact, the concern of safety was raised by female traders who were asked of the challenges that they are facing when they are conducting this trading, particularly safety issues in the field, because in the studied region, the need to collect firewood in the field has decreased. People tend to buy firewood that is even collected from other regions. There's no more need to fetch water in the field in the studied region. Therefore, there's fewer people in the field at a given time. So hence the traders that were asked, particularly females, expressed their concerns or when it comes to safety. So it's not one of those businesses that we can say is male dominated and are excluding females. It's open. There's not even a register of who's trading. The local authority which is leaded has no knowledge of who's trading and where. It's just a self-regulating kind of activity.
Thank you very much, Claude. I see we've basically run out of the scheduled time or the allocated time for the question and answer sessions. There's a few more in the Q&A box, most of which have been answered also. There's a few others, unfortunately about 3 or so that we didn't get time to address here. So yeah, a few with regards to the prickly pear trade, and Claude, you sort of touched on the perhaps local authorities or government can look at regulating it a bit more and trying to improve the conditions under which this is happening. So somebody also asked that, we know that prickly pears are invasive in South Africa, but don't you think if DFFE can allocate permits to farm sustainably and under controlled conditions that would help prevent the spread and also perhaps make it safer for people to do? Looks like we lost Claude. Okay, then I'm going to end the question and answer session here also. Just thank you again to all the presenters for the time and effort that you put into preparing very informative presentations. Also to the people who posed questions and participated in the discussion. And I encourage everybody, we heard of several very good initiatives in this session with regards to determining the impact of these alien invasives and how you as a general scientific and regulatory and government and just interested people can get involved in reducing the risks and just picking up the new occurrences and spread of these alien invasives. So please do utilize the opportunities that's out there and assist the scientists and the government bodies in collecting the information that's so necessary to manage these alien invasives. And also thank you very much to my co-chair sitting here opposite from me. Thank you very much for your assistance, Phumutso. It's been a pleasure to work with you. And to the organizers of the symposium, thank you very much. Then we're going to go to lunch now, and we will start up again at 1 p.m. sharp for the next session. Thank you very much, everybody, and enjoy your lunch.